in botany. Now I am discussing about some more important topics in genetics. Those are test cross, incomplete dominance, codominance. What are they? Test cross, incomplete dominance, codominance. These are the more important topics in genetics. First one is the test cross. So if you observe the topic name, the topic is the test cross. Here means to test something. The test cross means to test something. What is testing here? Genotype of F1 generation is testing. What is testing here? Genotype of F1 generation. We know that what is F1 generation. We already seen in previous videos. So what is test cross here? To know the genotype of F1 generation. For this, to know the genotype of F1 generation, we have to cross the F1 plant with the recessive parent. What we will do? We have to cross the F1 plant with the recessive parent. There are two parents are there, dominant parent and the recessive parent. So the F1 generation plant will be crossed with the recessive parent. If you take the one example for this, to know more about test cross, we have to take an example. The example is tall pea plant. The parents are parents. Tall pea plant crossed with the dwarf pea plant. In first linear generation, that is F1 generation, we know that 100% plants are tall. We know the phenotype of the F1 generation. What is the phenotype? Tall. But we don't know the genotype of the tall. Genotype of the F1 generation. But externally, this plant visible as tall. But what is the internal feature? We don't know. That is genotype. To test the genotype of these F1 plant, we have to cross this F1 plant with the recessive parent. Why this one is called as the recessive parent means already one parent character visible outside in F1 generation. That parent is called as the dominant parent. Another parent character does not visible outside. That parent is called as the recessive parent. Here we know that already the tall one is the dominant parent, dwarf one is the recessive parent. To test the F1 generation genotype, we have to cross the F1 plant with the which parent? Recessive parent. So if we take the symbolic form, capital T, small t, small t. Okay. So from here, capital T. From here, small t. These are the gametes. This is the unknown. We don't know. But this is the tall. We know that. But we don't know the genotype of this F1 plant. Okay. So which plant it is? Phenotypically it is tall, but genotypically it is don't know. Okay. So there are two possibilities are there for the tall. One is the capital T, another one is the capital T, small t. This is the homozygous tall. This is the heterozygous tall. For the genotype, this F1 generation plant has two possibilities. If it is, sometimes it may homozygous type, sometimes it may heterozygous type. We don't know that. Yes. To test this genotype, we have to cross this tall plant with the dwarf parent. So how it is possible? See. How many possibilities are there? Two possibilities are there. What is the first possibility? Capital T, capital T. What is the second possibility? Capital T, small t. They both are tall. They both are tall. Okay. So if the F1 generation plant cross with the recessive, small t, here also recessive, small t, recessive parent. So here, this is the unknown F1 generation plant. This is the unknown F1 generation plant. These both are crossed with the recessive parent. See, if this plant homozygous type, what will happen in F2 generation? If this plant heterozygous type, if it is crossed with the recessive parent, what will happen in F1 generation? We have to see that. Okay. So first of all, see, if we draw the Punnett square, capital T, small t, capital T, small t, capital T, small t, capital T, small t, capital T, small t. What is the phenotype ratio? Phenotype ratio is, what is this? Tall. This one tall. This one tall. This one tall. 4:0. What is the genotype ratio? What is this? Homozygous or heterozygous? Heterozygous type. What is this one? Heterozygous type. What is this one? Heterozygous type. What is this one? Heterozygous type. Same. 4:0. It means phenotype and genotype ratios both are same. When it is, if the unknown F1 generation is homozygous type. When the unknown F1 generation plant is homozygous type, phenotype and genotype ratios both are same. So, when the unknown heterozygous type, when the plant is unknown plant is, if it is heterozygous type, what will happen? See. Small t, capital T, small t, capital T, small t, capital T, small t, small t, C. So what is the phenotype ratio? Tall. This one tall. This one dwarf. This one dwarf. The phenotype ratio is 2:2. How many dwarf? 2. That is equal to 1:1. So what is the genotype ratio? Genotype ratio. Homozygous type. Sorry, heterozygous type. Homozygous type. That is equal to 2:2. That is equal to 1:1. Here also phenotype ratio, genotype ratios both are same, but ratios are different. Here phenotype ratio both are same, but ratios are different. If the plant is homozygous tall, if it is crossing with the recessive parent, the phenotype ratio is 4:0, genotype ratio is 4:0. If the plant is heterozygous tall, if it is crossing with the recessive parent, the phenotype ratio is 1:1, genotype ratio is 1:1. By this, we know that genotype of the F1 generation. We test the genotype of the F1 generation. This is the test class. So test class means to know the genotype of F1 generation. For this, we have to cross the F1 plant with the which parent? Recessive parent. This is the, what is the test class. Okay. The next topic is incomplete dominance. incomplete dominance. It means F1 did not resemble either of the two parents. Means what? The F1 generation plant did not show the two parental characters. It shows the other characters are in between characters or middle characters. That is incomplete dominance. So, for the explaining the incomplete dominance, scientists have taken the example of snapdragon plant. Which plant? Snapdragon plant, also called as the dog flower plant. Its scientific name is Antivinum majus. What is the scientific name? Antivinum majus is the scientific name of the dog flower or snapdragon. The snapdragon plants shows the two types of variations. Some plants use the red flower colors, red color flowers. Some flowers shows the some plants shows the white color flowers. Here, scientists are crossing the true breeding red color flower plant with the true breeding white color flower plant. See, which plant? Red color flower plant with the white color flower plant. So, these are the, what are these? Parents. The gametes are meiosis division, they splitting into gametes. Yes. Any one of the gamete from the red color flower plant, any one of the gamete from the white color flower plant. So, crossing. The F1 plant will be. What we are expecting in F1 plant? What we are expecting in F1 plant? Most of the students are expecting F1 plant is red, but that is wrong. So, what is the definition? F1 did not resemble either of the two parents. What is that? The F1 generation plant did not show the two parental characters, which shows the other characters, middle characters or in between characters. So, unfortunately here, capital R, smaller plant will be produced. What is the capital R, smaller plant is? Is it red or white? This is not red, this is not white, this is pink. Unfortunately, which color flower plant will be produced in a F1 generation? Pink color flower plants will be produced. The pink color is the character not present in two parents. Not present in two parents. The one parent is the red, another parent is the white. Unfortunately, which will be produced here? Pink will be produced. This is an incomplete dominance. So, if the red will be produced, if the white will be produced, that is complete dominance. It satisfies the law of dominance. What is law of dominance? Any one of the parental character act as a dominant in a one generation. That is law of dominance. Here, the incomplete dominance does not satisfy the law of dominance. That is why it is also called as the incomplete dominance. So, F1 did not resemble either of the two parents. It was in between two. Means the character in F1 generation knowing parents. So, in between parents, the middle of the parents, whatever it may be. If we self-pollinate the F1 generation plant, what happens? If we draw the pollinate square, capital R, small r, capital R, small r, capital R, capital R, capital R, small r, capital R, small r, small r, small r. Can anyone guess the phenotype ratio, genotype ratio in F2 generation? This is the F2 generation. Can anyone guess what is the phenotype ratio? Which plant it is? Red. This one? Red. No red. This is pink. See, capital R, small r. What is this? Pink. So most of the students thought like this is a red, but that is wrong. This is a pink. This one pink. This one white. How many phenotypes here? 3 phenotypes here. One is the red. Second one is the pink. Third one is the white. The phenotype ratio is the red is to pink is to white. That is equal to 1 is to 2 is to 1. Yes. Unfortunately, the phenotype ratio is the 1 is to 2 is to 1 in incomplete dominance. Can anyone guess the genotype ratio here? Genotype ratio. Homozygous. Homozygous. These two are heterozygous. This one homozygous. Homozygous red. So heterozygous pink. Heterozygous pink. Homozygous white. Then that is equal to how many homozygous red here? 1. How many heterozygous pink here? Is to 2. How many homozygous black here? Is to 1. Yes. See, what is the phenotype ratio? 1 is to 2 is to 1. What is the genotype ratio? 1 is to 2 is to 1. As like same in testicles. In testicles also, phenotype ratio, genotype ratio, both are equal. Incomplete dominance also, phenotype ratio and genotype ratio both are equal. So this is an incomplete dominance. Why? Because it is called as incomplete dominance. The F1 generation plant does not show the parental character. That is why it is an incomplete dominance. Okay. Our next topic is codominance. Codominance means the F1 generation plant will be produced or will be shows both parental characters, which is called as the codominance. The F1 resembles both parents. Dominant and recessive characters both are visible in F1 generation. That is codominance. Co means mixing. The characters are mixing together, dominant. That is codominant. To explaining about the codominance, example is lentil seeds. The example is lentil seeds. Some lentil seeds shows the spots and the seeds. Spots and the seeds. Such seeds are called as the spotted seeds. CS. C means codominance. CS. Dotted seeds. Some lentil seeds shows the dots on the surface of the seed. Such seeds are dotted seeds. CD. Lentils. Here, spotted seeds. Lentil plants are crossing with the dotted seeds. Lentil plants. Like this. This is the crossing. CS spotted seeds. CD dotted seeds. Gametes divides into gametes. In F1 generation, CD will be produced. What you are expecting here? Is it a spotted plant or a dotted seeds plant? Exactly. The plant shows the both characters. The seed shows the both characters. Some dots are there, some spots are there. Both parental characters visible in F1 plant. If we self pollinate the CS, CD, CS, CD, CS, CD, See, which plant is spotted plant? This one spotted and dotted. This one spotted and dotted. This one dotted. So how many plants are phenotypically produced? Three types of plants. What are they? First one, spotted plants. How many? One. How many spotted and dotted are there? Two. Spotted and dotted. How many? Two. How many dotted plants will be produced? 1. That is equal to, what is the phenotype ratio? 1 is to 1. Yes. What is the genotype ratio? See, homozygous spot. Heterozygous spotted and dotted. How many? 2. Homozygous dotted. How many? 1. How many homozygous spotted are there? 1. That is equal to 1. How many heterozygous spotted and dotted are there? 2. Finish. Last one. Homozygous dotted. How many are there? 1. What is the phenotype ratio? 1 is to 2. In test class, in incomplete dominance, in codominance, in 3 topics, in 3 types, the phenotype ratio and genotype ratio both are same. So, in test class, phenotype ratio and genotype ratio both are same. In incomplete dominance, phenotype ratio is the 1 is to 2 is to 1. Genotype ratio is the 1 is to 2 is to 1. In codominance also, phenotype ratio is the 1 is to 2 is to 1. Genotype ratio is the 1 is to 2 is to 1. So, these are the topics we discussed today. Okay.